Hi everyone, welcome to my channel Srinivasa Biology Tutorial. For this section to discuss class 11 biomolecules part 3 next, that is after nucleic acids. After nucleic acids, what are the nucleic acids? Nucleic acids nothing but as an either like as in a DNA and then RNA. DNA and RNA is called as what? A nucleic acids. Now see that one. Here the other type of in a macromolecule, that is one would there find acid soluble fraction another living tissue, the nucleic acids. So another living tissue, their insoluble fraction is called as what? Nucleic acid. These are in a polynucleotides together within a polysaccharides and polypeptides. These are compressed to the true macromolecular fraction, the living tissue of the or cell. So you take as in one particular living tissue, in that living tissue they having as in polysaccharides as present as well as in polypeptides as present. They having as polysaccharides as well as polypeptides as present. In these two both they gather to get form as in one macromolecular fraction is present in that in living tissue and then or cell. For example, they given for the nucleic acids, the building blocks of the nucleotide. The nucleotide has in it that three chemically distinct compounds as present, like as in a what? Nitrogen base and pentose sugar and phosphate group and then phosphate group. So these are the three compounds as present. And one heterocyclic the compound and second the monosaccharide and third is in their phosphoric acid and nothing but as in phosphate. So these are the completely in the building blocks of the nucleic acid. Now as you notice the figure 9.1, you see as in a figure the figure 9.1 like as in a here the heterocyclic compound, the nucleic acids, they are nitrogenous base named as in adenine, guanine, uracil, cytosine, thymine, adenine, guanine, and the substance the purines, while they are rest the substituent the pyrimids. So some they having as a purines as present here purines. Here purines, and pyrimidines. Here they are substituted by as in pyrimidines. Their skeleton hydrocyclic ring is called as in purines, like and pyrimidine respectively. So in this total DNA consists of as what? They consist of as in purines as present, and then they're complete their opposite they bind with as what? Pyrimidines as present. They complete as what? Pyrimidines as present. The sugar found in the DNA polynucleotide either in a ribose in that present as in a monosaccharide pentose sugars or in a second deoxyribose. So it is in a ribose sugars. Now the nucleic acid contained as a deoxyribose is called as a deoxyribonucleic acid, that is in a DNA. So RNA nothing but as what? Ribonucleic acid consists of what? In a the second the deoxyribo group is present, and while this which are contained as in a ribose is called as in a RNA, that is in a RNA. Okay, this is in a completely in that DNA. Okay, next to discuss in that in a structure of proteins. Now to discuss as what in a structure of proteins. See, in the structure of proteins, the protein is mentioned as an earlier the heteropolymer contained as in the straining the amino acid structure, the molecules, the means differ, the different things, the different contents or different cortex, the inorganic chemistry. So here proteins they have different structures, like as in that in a structure variable referred to as any molecular formula, like as in the sodium chloride, magnesium chloride, etc. So here different proteins has and different structures as present. Now the organic chemistry always write about as a two-dimensional views of the in a molecule, while then representing the in structure of the molecules, like as in that benzene and naphthalene, etc. So they have forms as in different structures also. Okay, like as in benzene and naphthalene. The physicists that they configure up to as in a three-dimensional views the molecular structure, where the biologists they describe as in the protein structure in the as in 4 levels. So in physics 3 levels, and here in biological view in that under the proteins has in 4 levels we get described. The sequence of the amino acids, like as in first the positional information and protein, which is the first amino acids, which is second and soon on this called as a primary structure. So when they get formation of directly in one particular structure to get formed, that is called as a primary structure of protein. Of a protein is the meanified as a linear, the line, the left end represented by as in that first amino acid, the right end to represent as in that last amino acid is present. So it is in the first amino acid, here they completely has in what in a last amino acids will be to get formed. Now here the first amino acid is called as in that N-terminal amino acid. The first amino acid is called as what? For example, they given here. So here it is in a first amino acid, it is in a what? N-terminal amino acid. The last amino acid is called as what? C-terminal amino acid. That is in a C-terminal amino acids. Now here protein, the thread do not existing the throughout as in the extended grain rod. So it is complete to get forms as what in grain, the rigid rod-like structure. The thread folded as in that for a helical or they revolving the strain cases. So of course that in some the portion of the protein, the thread, the arranged as in a form as in a helix. Some they get arranged as what in a helical structures, alpha helix and beta helix to get forms here. Alpha helix and beta helical structures also to get produced in the proteins. In proteins in right hand, the helical observed as in that, otherwise the protein, the thread folded as in that for what is called as in the secondary structure. So in that secondary structure also to get formed in the secondary structure to get helical structure to get produced. It is a one hand, it is in a starting, it is in the end of that in a protein form. Okay, in addition of the long polymer chain here, in the addition of long protein chain is also folded up to as in a itself like hollow woolen ball, it given as rise to as in a tetracyclic protein structure. So it is to get formed as in a the complete woolen ball like structure to get forms as what in a woolen ball like structure, that is called the tetracyclic protein structure. This is given as a three-dimensional view of the protein. So the completely the tetranary structure to get formed as in that three-dimensional protein structure. And tetranary structure is also absolutely necessary for as in the mainly biological activities of proteins. So the biologically protein structure is mostly used for as in a this tertiary structure. This is a tertiary structure. Now in some proteins and that in assemblage for more than one polypeptide or in a subunit. So more than 2 or 3 subunits as present, the manner that while the individual folds the polysaccharides, the subunits the arranged with as in respect to as each and another. So that in a spherical or space the arranged up to as in each and other from as in the cube-like structure to it is to complete to get form as in a one cube-like structure. So this adjuncture of the protein the otherwise is called as in a quaternary structure. So when we are completed to get form as in a one quaternary structure like proteins, this is called as a quaternary structure. So of these proteins, the adult human hemoglobin consists of as in a 4 subunits. So when you take as in a that human blood group has in a that 4 heme groups as present. The complete has in a what in a 4 heme groups as present like. Okay. Now the two otherwise in identical and each another, and two other subunits in alpha and two subunits as in a beta together to form as in a human hemoglobin HB. So one is in alpha and another what beta, the globins as present. So this is in a quaternary shape of structure. Okay, these are the completely in a protein and protein structures. Okay, these enzymes. Enzymes are almost called as proteins. Here in some like as a enzymes. Here very important point is, what is that one? Almost enzymes are proteins, but proteins are not enzymes. Here enzymes are called as any proteins. Enzymes are called as what? Proteins. But here proteins are not called as enzymes. This is very, very important. Okay, here saying here the nucleic acids behave like as any enzymes. These are called as any ribozymes because they are ribozymes already, you know, the ribosomes are mostly to participate as what? In a metabolic activities. Am I right? Yeah. Here can be the enzyme can be their diagram. The enzyme like as a protein, the primary structure, that is an amino acid sequence of the proteins. So the proteins, the mostly enzymes, they having as in a one specific the sequence of in a protein is present. Now that enzyme like as in a protein have the secondary, the tertiary structures also present. The enzyme, the protein part is present. In that protein part, the mostly consist of as in a primary and secondary parts also they get present. Now you look as in the tertiary structure, you will be the notice the backbone of the protein chain, the fold up to itself. It is a chain. It look like a the crisscross itself, and then hence to make the cell splits or in a pockets they're made up of. See, when you take as a one particular enzyme, here you take as a one particular enzyme. In this enzyme, they consist of as what? In a some pockets. They have consist of what? Here some pockets as present. These are complete called as in a their pockets. These are called as what? Pockets as present. See, they are crevice or pockets they are made up of. Here, such pockets they are called as what? Active sites. See, these pockets we are called as what? Active sites. Now the active sites are being the enzyme that in a crevice or pocket in which has the substrate fits. So in that active site they get bind with as what? They bind with as in a substrate. See, this is in a substrate. Is what? In a substrate. So substrate to get bind. For example, this is an active site. Am I right? In this active site to get bind with as in a substrate to get bind, then get what? To get formed as in a product. Okay? Line. Here, thus enzymes to gather to active site, the catalyst, the reaction, the high rate. So he's saying about as what? When the enzyme active site to get substrate to get bind, here what happened? The enzyme activity is complete to get more. They having as what? They get more. Now the enzyme catalyst to differ as an inorganic catalyst, maybe they always to, but the differences needs the mention. So here all enzymes to get not perform as in the same function. The different the enzymes to get perform as in different functions like the inorganic catalysts work efficiently at high temperatures and high pressures, while the enzymes to damage high temperatures above the 40 temperature. So just I say that one only. What? Enzymes perform as in different functions depends upon depends upon the factors. For example, see here mostly the enzymes to get damaged to a high temperature above the 40 degrees. So in the factors are the favorable. What happened? They performed as in the good functions. Like okay. Now, however, the enzymes are isolated from as in organisms who are the normally to under the the extremely high temperatures. Like example they given here hot wines and the sulfur springs. So here some bacteria or some enzymes to get grow as in the heavy temperatures or heavy regions also like. Okay. Now the stables that retain that catalyst the power even the high temperatures up to 80 to 90 degrees. So the thermal stability is as an important quality such as the enzymes isolated for as in the thermophilic organisms. So here like as a Taq polymerase, when you see as a Taq polymerase, this Taq polymerase enzyme to get grow around as an 80 to 90 degrees the temperature is also. Thermus aquaticus enzymes. Am I right? Got it. Remaining to discuss in the next class. Thank you for watching. Please share, subscribe and like.